Hello, I'm Luca Comai. Let's continue with the 10 most important features of meiosis. Number 2, in preparation for meiosis, chromosomes duplicate. Here is a schematic view of a chromosome where the blue line represents a double-stranded DNA molecule, the small circle, the centromere, abbreviated as CEN. The centromere divides the chromosome into 2 chromosome arms. In the S phase of the cell cycle, as it happens for mitosis, the DNA replicates, forming small bubbles that grow at their ends until the whole chromosome is made of 2 identical double-stranded molecules called chromatids. Chromatid cohesion, that is the tight attachment to each other, is mediated by a protein complex called cohesin. The replicated chromosome enter G2 phase. It is important to understand that the replicated chromosome is still one chromosome made of 2 chromatids. I prefer not to draw a replicated chromosome like an X. Number 3, during prophase, chromosomes condense and at the same time homologous chromosomes pair. Homologous refers to the 2 copies of a given chromosome. Their sequence is very close or identical, and they carry alleles of the same gene. Pairing involves the side-by-side alignment of the 2 replicated chromosomes. The red chromosome on top is made of 2 red chromatids, and the blue chromosome at the bottom is made of 2 blue chromatids. Concurrent to pairing, double-stranded breaks are introduced by endonucleases at many chromosomal sites. Even if technically in pieces, the chromosomes are still held together by proteins and do not fall apart. Most double-stranded DNA breaks are repaired, that is, resealed, but some initiate meiotic recombination, a process that involves the DNA strands of one DNA double helix separating and invading the homologous chromosome DNA. The DNA strands cross over, forming a structure recognizable through a microscope and called chiasma. It means cross in Old Greek. The plural is chiasmata. DNA in chiasmata is cut and spliced to form recombinant chromatids. These 2 chromosomes, for example, have exchanged a piece of the left arm. Note that 2 chromatids are recombinant, formed by joined red and blue DNA, and 2 are parental, formed by the original DNA color inherited from one parent. Number 4. Each chromosome pair engages in 1 to 2 crossing overs, which typically occur between chromatids of the 2 homologous chromosomes, and not between the chromatids of the same chromosome. Interestingly, a crossing over that is forming inhibits formation of other crossing overs nearby. This is represented by the T-bar, a symbol indicating repression or inhibition. This phenomenon is called crossing over interference. What is the purpose of crossing overs? First, crossing overs entangle the 2 homologous chromosomes. When the spindle apparatus attached to the centromeres tend to pull the 2 homologs apart, the crossing overs resist the splitting of the 2 homologs and are important for ensuring the correct placement of the paired homologs on the meiotic metaphase plate. As an aside, note that the 2 chromatids of each chromosomes remain glued together until late metaphase 1. Second, crossing over recombine the parental chromosome, creating new allele combinations. In this example, the F1 of the 2 inbred parents, the red and the blue, recombines parental chromosomes to form gametes that may contain recombinant or non-recombinant, that is, parental chromosomes.